Hi everyone, and welcome. So I'm really happy to be here and to present Giotto Suite, which is the successor of Giotto, a previously developed spatial transcriptomics pipeline. And so first of all, a quick recap of what Giotto exactly did. So Giotto was a pipeline that could work with any type of spatial transcriptomic data. And so the raw data of spatial transcriptomic data is the image itself. And so the way we approached it previously was that we said, let's just try to make it as abstract as possible. And so the only thing you would need was basically a gene by cell matrix. And then the other thing would be the actual cell coordinates. And so with those two, you could make a spatial network or a spatial grid, and that would be sufficient to analyze all sorts of different spatial technologies and spatial platforms and look for spatial gene expression patterns, cell-cell communication. You could also do sorts of visualizations. However, there were definitely a couple of shortcomings that we found out when we were developing Giotto. And I think one of the biggest pitfalls and challenges comes from the fact that there's a clear disconnect between the actual technology and the imaging part and the data analysis. The data analysis is probably more similar to single-cell RNA-seq, while the technology or the imaging is its own little world, and there's very little overlap. So a couple of things I noticed is that, of course, the data set, size, dimensions, and format can be very, very different. In addition to that, and this is perhaps even more important, we only use a fraction of the data of the raw imaging data. In fact, a lot of the imaging data is often not used. Let's just think about cell morphology and everything else that is kind of like surrounding this. By definition, spatial transcriptomic data is also multimodal. You always have like the image, and then on top of that, you have spatial transcriptomic information. And since we are working with spatial data, it by definition should be interactive, which we can use our own eyes to help us guide what are the interesting regions, what are the interesting patterns. And so today I just want to briefly touch upon a couple of new additions that we add in Giotto Suite to make it a lot better compared to our previous Giotto version. So first of all, if you think about the input, you typically have like one image, but if you have like a Z-stack or you have a 3D data set, you have multiple different images. On top of that, you maybe want to overlay your results with other type of information, for example, the H&E staining or any type of immunofluorescence staining. So you need to have like this typical image processing task. And stitching is typically very straightforward, but something like registration might already be a little bit tougher. And so really to go from the image to the spatial transcriptomics part, to make that happen in one go, we also implemented registration. And just to kind of like to show you why it is important. So what you would have is if you have a tissue block and you have like 7 consecutive slices, it would be very hard for you to manually overlay them, but you can do something it's called registration, and that is basically aligning all the different slices right next to each other. And so what happens then, if you look at the spatial coordinates of all these different spots or cells within each slice, so there are 6 slices, and here after registration you can actually see that they're nicely aligned. And that actually makes sense because if we look at the cell types, the predominant cell types in each spot, then what we typically see in a 2D slice is that their homotypic interactions, so interactions between the same cell types, are more frequently observed compared to heterotypic interactions. However, this is a little bit distorted along the z-axis, and so what you can do, so once you do the registration and you look at how is these, what can we learn now, and what is different is that we see that those homotypic interactions where 1 interacts with 1, 2 with 2, and so on, they are typically increased. So this is what you see here over the diagonal. So this indicates that now also along the z-axis you see that there is this homotypic interactions. So that definitely makes sense. Perhaps the biggest addition is the usage of segmentation, or we allow a way to store and use segmentation results. So what happens is that you normally have your tissue of interest or your image of interest, and then you can use all sorts of different technologies, and I'm just highlighting a couple of them, but we made it so that we can accept almost all the different outputs that they generate. And so what you would have is something like this, which is typically a mask file. You can have that for multiple different layers, for example, a nucleus, cell membrane, but that can go up to as many staining as you have. So the way you represent it is we think about it as the fact that a tissue by itself is inherently very hierarchically organized. You have the multicellular components, you can go to the cell-cell components or the cell, single cell, and then within the cell you have the subcellular components. And so what you can do is you can use any of those segmentation tools and you identify them, and we can convert them into polygons, which represent a very efficient way of using that in any type of downstream spatial analysis. On top of that, we can link that in different planes so that you have a very hierarchical presentation of your cell in a very memory efficient manner. And so on top of that, if you now start integrating the actual transcript locations here depicted by all the different colors, you can also do all sort of things that were not previously possible. And just to give you like two very little examples, for example, you can do quality control based on what is the segmentation effect. So here you have all these cells that are segmented, and now the dots in red represent the dots are represent as the transcripts that are not assigned to any cell. So that means they are either your segmentation is either bad, or you're interested in knowing like what are those transcripts that are not found within a certain cell boundary. Something else that, in a very simple thing you could do is you could identify where are those transcripts localized. So they might be within the cell boundary, but some of them might also be within the nucleus. And so using that information, you could use that to identify what are the transcriptional spatial dynamics. Something like velocity, but there's a lot more that is possible. So I'm gonna now move to the next part. So this was the representation part, which is probably the biggest part. But there's also, like, as I said before, there's a very interactive component, and I think the analysis by itself should be interactive. So there is a way, we are working on making this human-guided exploration more easily. So for example, you have this spatial plot and you identify an interesting region, and then you can quickly do a lasso around it, identify it, you move that back to the Giotto object, and you can use that in your more interactive R script analysis. We are also working with the spatial community to make it easier. We have interoperable structures for a number of other spatial toolboxes, and we're also heavily interested in using Binder as a very simple way to allow people to test Giotto in a browser environment. So you can simply launch Binder within your browser. You don't have to install everything. Under the hood, there are a number of packages involved. We created a large number of notebooks with all kind of like different examples, and so you can open that one, and then you can run it, you can share results with other people, and if you like it, you might want to install Giotto on your local machine. And the last part is we're also working together with other developers, and we also invite everyone that thinks they might have an interesting idea or method, and they want to see this integrated in Giotto. And so what we're doing to help facilitate is we're kind of like splitting Giotto into multiple different like subcategories. So smaller packages, for example, we have the Giotto class itself, the Giotto images, the Giotto utilities. So that it's very easy to extend one of them or like edit one of them without breaking anything else. So with that, I just want to say, so Giotto is a really big project that is, and we do it together, or my lab, Dries lab, does it together with GC's lab at Mount Sinai. There are a lot of people involved and helping us, but here for this particular talk, I just want to highlight, and I want to thank George and Muzamil and Natalie from GC's lab. And thank you all.